Hello, everybody. I am just back in the office after going to the American Ornithological Society annual meeting up in Anchorage, Alaska, where I presented this talk. And what I want to do is record it now so that anybody can look it over and see what we found, because this is kind of the interim report on the Acadia Bug project. So I was in a disease and ecotoxicology session, and this is a bird conference, remember, so this is a little bit bird focused, but I end up talking about bugs a lot. So the title of my talk was Cross-Ecosystem Subsidies in Acadia National Park, How Invertebrates Can Help Us Understand Mercury Exposure in Songbirds. And I want to start out by acknowledging all the different people who came together and funding to make this project happen. So I'm combining some data that we collected in Olympic a while ago and data we collected in Acadia in 2012 and some dragonfly mercury data that's publicly available online. And so all of these funders were also huge and helpful. But most important were all of the undergrads who came with me to Acadia last summer to help collect these data. Many of which you'll see as the slides go may use this data in their senior research projects, and so I really couldn't have done this work without all of these guys. So I want to start, I was kind of the lead off ecotox talk in my session, and so I wanted to start off just talking about wildlife toxicology in general and the fact that it's still a relatively young field. So Silent Spring was kind of our first look in 1962 at, hey, maybe we're causing problems with the type of chemicals and things we're putting into the environment, and they could have an effect on wildlife. But even recently, the vulture crisis in Asia showed that we can still put things into the environment that we don't realize are going to cause harm, but then cause huge widespread declines. So the problem with the field of toxicology is that it's usually reactionary, not precautionary. And I mean by that you find an animal or a group having problems, and then you kind of race to assess what's going on, but we're not usually getting too ahead on looking forward and being precautionary. So one of the problems is that you have to get people to care about being more precautionary. I study mercury, and I even think mercury in fish and wildlife can be hard to care about. Most of the effects are sublethal. There's not always a clear point source that we can clean up, but at least it does have human health implications because people eat fish and eat some of the wildlife that are affected by mercury. But mercury in songbirds, which is what I really study, is super hard to get people to care about. They're not a game species, so no human is eating them for the most part, in the United States at least, and it's hard to quantify how they're impacted by mercury, and they have a lot of other maybe bigger threats. The whole conference kind of that I was at covered all these different things that are threats to birds, climate change and deforestation and disease and all these things. And so of course, like, this is just one of those many threats that can affect songbird populations. So to combat this apathy and get people kind of thinking about what contaminants do in our environment, the USGS, Park Service, University of Maine came together to create this dragonfly mercury project where they got people out into the field with them to collect data by catching dragonfly larvae in all of these parks across the United States. And so it's a really awesome way to get people interested in both land preservation and contaminants and just kind of understanding what's going on beneath the water. So the question that I've had since the Dragonfly Mercury project started is, what are dragonflies actually sentinels for? I think they're good sentinels for fish, and so human health. But are birds really eating dragonflies in super high numbers? A lot of the birds that I study are in the 10 to 20 gram range and not necessarily the fastest things, and so eating an adult dragonfly might be pretty hard for them. They're very big and fast. But it's very likely that our birds eat a high number of other emergent aquatic insects like mayflies and stoneflies that emerge, live shorter amounts of time, and don't move quite as much once they emerge. And I think those bugs are probably really important food sources for riparian songbirds. So the Dragonfly Mercury project had these paired coastal parks where they did intensive sampling in Olympic and Acadia. So in 2014 and 2015, when I was doing my PhD out in Oregon, other people the Dragonfly Mercury project sampled dragonflies, and I went out and sampled songbirds at the same sites in Olympic. And so what we wanted to see was how does a dragonfly geomean compare to the songbird geomean at the same site. So we had all these sites that we sampled together, and when we plot them against each other, we see some sort of correlation between dragonfly mercury and songbird mercury, but it's pretty weak and hard to say that dragonflies predict songbirds in any real way. So why don't they correlate? Dragonflies are top predators and long-lived, so they might not reflect the prey source that the birds are actually eating. Like I said, they might be eating more of the other types of emergent aquatic insects. And birds can also eat terrestrial prey, so they can be eating caterpillars and butterflies and things that never had a tie to the aquatic system. So once I had done the work at Olympic, I really wanted to get out to the other park, Acadia. Unfortunately, but fortunately for me, I got a job, so I got delayed. And so the Acadia sampling is a little disjointed, which we'll have to deal with, where the dragonflies were sampled from 2014 to 2016, and then in 2012 and 2018, we collected songbird blood samples. So these are all the sites where dragonflies were sampled at Acadia. You can see there's a lot of variation in their mercury levels. And we decided to focus on 4 sites in particular that kind of have a range of mercury levels. And more importantly for my project, they had easy access and we could take citizen scientists in there easily. Because what I wanted to do as part of my Second Century Stewardship Fellowship was correlate songbird mercury to dragonfly mercury at all of the different dragonfly mercury project sites in Acadia. And then also on those 4 target sites, track changes in insect phenology using both researcher methods and then also bring citizen scientists out to get them interested in this project. And then also assess what the birds are eating using next generation sequencing of fecal samples. So the idea was that we'd be there all of June and July for 8 weeks. Each week we'd collect bug and water chemistry at each site. Every other week we'd bring citizen scientists out to do bug sampling at each site. And then right at the peak of the breeding season, we would do songbird blood sampling to get an idea for what bugs were there before and after we sampled songbirds, which is pretty much their breeding season. Unfortunately, I'm not going to talk about this because there wasn't time, so there's going to be another presentation about the citizen science effort at a later date. So I'm going to talk mostly about how our researcher bug and water chemistry sampling influenced or correlated with songbird blood. Also, unfortunately, right in the middle of songbird blood sampling, I broke my ankle, and I was the only one who could do a lot of that work, and so we didn't get to quite as many sites as I had hoped, which was a huge bummer. But I'm going to show you what we have, and the hope is that someday we'll get back to these sites and get a better idea about them. So since it was a bird conference, I felt like I had to at least show some of the birds that we were collecting from, because the important thing here is that these are not tree swallows or anything that's obligate to riparian systems. A lot of them, like the yellowthroat and the song sparrow, do predominantly occur in aquatic or riparian areas in Acadia, but the other birds can occur in lots of other areas, uplands, where they could be eating other types of bugs besides emergent aquatic insects. So after this, I'm just kind of going to group all of the birds we caught at each site together and just look at, in general, bird mean mercury concentrations. So when I kind of compared them all together, so this is the same graph but now looking at Acadia, where I took site, dragonfly geomean, and songbird geomean. The correlation is even worse at Acadia. There are some issues with how I did this, where the dragonflies and the birds are the same year. Gilmore and Aunt Betty is not in exactly the same location in the watershed, so that could be some issues going on with why they don't correlate well. But I don't think those issues explain these huge differences in that you can see here some really high dragonfly sites that have really low songbird levels, and some really low dragonfly sites that have pretty high songbird levels. So it's important to kind of understand these study sites and dive into them. So we sampled intensively these 4 sites across Acadia. Gilmore Marsh is upstream of Aunt Betty Pond. It's kind of a wide, open, shallow marsh. Marshall Brook is on the quiet side of Acadia, and it is kind of degraded area that they're hoping to restore, so we wanted to do more work there. The Schoodic Beaver Pond is a really small kind of wetland complex on the Schoodic Peninsula over here. And then Hunter Brook is a fast-flowing, clear stream that dumps right into the ocean over here. So an important thing is that these sites varied very considerably in water temperature throughout the season, probably based on kind of the morphology of their watersheds. So you can see that Hunter, for the most part, stayed pretty cool all summer. Gilmore really had some of these crazy spikes during really hot times, and in general went up. Same with Marshall and Schoodic, but they stayed at relatively lower levels. And I think this has to do with the type of stream that they are. So Hunter is forested throughout its reach, and it has a lot of cold, fast-flowing water. The rest of these kind of have wetland systems that allow them to get much hotter during the summer. So what we wanted to look at was how emergent aquatic insects changed and how they came out of the water. So what we did is we put out these emergent insect traps which floated on top of the water, and we're supposed to collect bugs as they emerged. So there's lots of caveats on this because we had trouble keeping these traps upright throughout the season, and so I tried to show just the data that I felt really confident about. And what you can see is not necessarily a peak of emergence at any one time, but in general, what I think is happening is that the sites vary in the biomass that they kind of output through emergent aquatic insects. And so we compared this to riparian terrestrial insect abundance. So now these aren't bugs that should be emerging from the water. We went into the riparian zone and collected them in small quadrats right around the river, and then could compare how their number of individuals changed throughout the season. So we had beetles, spiders, and hemipterans, which are true bugs, and we see not any sort of, at any of the sites, clear trend in increase or decrease in these different orders over time within the part of the season that we were measuring them. But the big thing that I wanted to kind of call out was in those terrestrial samples, we also caught emergent insects when we were collecting in the riparian zone. And what we can see is that, so you can kind of think of these terrestrial samples as what was kind of available to the birds at the time. And when I break it down by whether the bug that we collected was an emergent bug or a solely terrestrial based bug, we can see that these sites vary in how many emergent bugs they had in each of our samplings. So you could see Gilmore seemed to have a lot of emergent bugs that were collected in our terrestrial samples. Schoodic had kind of a peak early, Hunter relatively low, and Marshall really low. This meant there was a whole bunch of zeros in that data, where we didn't, in our terrestrial samples, got plenty of terrestrial-based bugs, but hardly any emergent bugs. So what I did is just looked at how sites vary in overall aquatic insect emergence. So now this is just showing number of emergent insects collected per day, and we can see that Gilmore has really high numbers and a lot of variation. Hunter has a little bit less, but still plenty of emergent bugs. Schoodic is a little lower, and Marshall is really low, hardly any emergent bugs coming out of Marshall. And so this is kind of really preliminary work, but we wanted to see, do birds vary in what they eat at each site? So we had done one round of next generation sequencing, and it's continuing this summer that Body is working on it. But what we think we're going to be able to show is in the individuals at each site, how many of these emergent aquatic insect orders are showing up in their actual diet. So there might be site-specific differences in what they're actually eating at the different sites. But stay tuned, there's a lot more going on this summer with this part of the project. So why don't the dragonflies correlate to songbirds? I think it might have to do with this emergence issue. So my general thought is, if you remember that these four sites had really different emergence, if we kind of look at it, we can say, okay, well here they had low mercury in the water, but really high emergence, which meant that a lot of low mercury bugs were getting out into the terrestrial system to be able to be eaten. Schoodic here had really variable mercury bioaccumulation and medium emergence, so it created kind of a wide range in what could be happening. And then down here, which was Marshall, the water and the aquatic system had high mercury, but it had such low insect emergence that this could be why the songbirds seem relatively protected or having relatively low mercury concentrations, because the stream was so degraded that it didn't have a lot of emergent prey coming out of it. So in general, what this is going to look like is that we can think about this in kind of a risk type scenario, where if you have high insect emergence and high mercury bioaccumulation in the aquatic system, the birds are at high risk. If you have low insect emergence and low mercury bioaccumulation, they're at low risk. But in these other quadrants, you don't really know. They could be at risk depending on how many aquatic bugs they're eating. There's lots of other factors at play to think about. The time of year that these bugs emerge. I wish we had been in Maine longer to kind of get the beginning and the tail end of emergent time. And if the birds choose to eat them or not, which we're hoping our metabarcoding is going to help us understand if the different individuals and different species are eating them, eating different things. So in general, remember the abiotic factors at each site are very different. There's no huge pulse in emergent prey at a particular time of the summer, at least between June and July when we're studying. It appears that the overall amount of emergent aquatic insects varies by site. This emergent flux may explain discrepancies from dragonflies to songbirds, because if the emergent prey isn't available for the birds, they don't have it to eat. And this creates a complicated risk based on mercury and insect emergence. So you can still, I'll post this on our Acadia Bug Project website, and you can follow the lab on Facebook, Instagram, and Twitter. And thanks for listening if you made it through this far. I would have been cut off ruthlessly if I was at the conference because I talked over, but hopefully you learned something and got to see our preliminary data. Shoot me an email if you have any questions.